Okay, so I'm explaining synaptic transfer. So basically what a synapse is, so here's a nerve cell, and so when the neural signal goes this way down the cell, the signal has to transfer from one nerve cell to the other nerve cell, and it crosses this area called the synapse. And so the synapse is the physical gap between the presynaptic neuron and the postsynaptic neuron. And the action potential goes down this way, down the cell, and when it reaches here, it will trigger a chemical release to continue the signal to another nerve cell. So when the action potential reaches the axon terminal, it triggers the opening of voltage-gated calcium channels. So here it triggers the opening of these channels, and then the calcium ions diffuse into the cell and promote the fusion of vesicles. So vesicles form around the neurotransmitters. Each vesicle can contain around 1,000 neurotransmitters. So the neurotransmitters are then released from the axon terminal through exocytosis. So the membranes open up and the neurotransmitters are in the synapse. The neurotransmitters then bind to the appropriate neuroreceptors on the postsynaptic membrane, which open ligand-gated channels. So depending on the neurotransmitter, they can increase or decrease the likelihood of the postsynaptic neuron to have an action potential. So the difference is some are excitatory neurotransmitters and some are inhibitory neurotransmitters. The excitatory neurotransmitters, or non-adrenaline, open up ligand-gated sodium channels, which is depolarization, and inhibitory neurotransmitters open ligand-gated potassium or chlorine channels, which is hyperpolarization. The combination of these chemical messages received by the dendrites on this side determines whether the threshold is reached for an action potential in the postsynaptic neuron. So the neurotransmitters that were used here, then some of them drift away by diffusion. Some are retaken by the presynaptic neuron through these pumps, which can be used again for another nerve signal. And some are broken down by other enzymes, so they can be reused for other